Thanks for the introduction, Wade. So today I'm going to be talking about a DNA metabarcoding assay for early detection of spotted wing Drosophila within unsorted bulk trap catchers. I do apologize to any taxonomists out there. I do know that this isn't actually a diffridid, and I'm in the diffridid section, but anyway. So first off, I want to acknowledge that I'm presenting from the traditional lands of the Wurundjeri people, and the fieldwork for this project was conducted in the lands of the Yorta Yorta and Bunurong people. I'd like to pay my respects to elders past and present, and any Aboriginal or Torres Strait Islander people who might be in attendance today. So a bit of background about spotted wing Drosophila. It's been an emerging global pest. It originated in Southeast Asia. And then, as you can see on this nice little infographic, it's started spreading throughout North America and Europe from 2008 onwards, and it's become quite established there, and also recently been detected in Africa. Now, unlike a lot of the native Drosophila we get in Australia, this is actually a pest of crops, and that's because it's got this serrated ovipositor that allows the females to lay their eggs in ripe fruit while they're still on the tree. Now, there was some great recent modeling done by James Mayno at CSER, looking at what would happen if spotted wing was introduced into Australia. And you can see there's a lot of chance of establishment and spread down the East Coast. So this shows that if we do get an introduction, we need to detect it early if we're going to have any hope of containing and controlling it. So early detection surveillance for spotted wing Drosophila is generally done with traps containing attractant lures. Now, there's no pheromone available for this pest, so what's used is apple cider vinegar or synthetic lures based on the volatiles from fermenting fruits. And also there's an alternative surveillance method that involves crushing fruit and floating it in a salt solution to release larvae, which then can be collected. However, the problem with both of these methods is they're not particularly selective, and they do catch a lot of other Drosophila and other bycatch insects. And looking for a rare specimen of spotted wing Drosophila in these bulk samples can be like finding a needle in a haystack. Traditional diagnostics would sort through this haystack piece by piece and identify each specimen using morphological examination or DNA barcoding. But the problem is when you have a lot of haystacks, maybe only some of them contain a needle, maybe none of them contain a needle. It takes a lot of time of our PhD level taxonomists to actually do this grunt work of sorting through all the samples. So at Agriculture Victoria, we've been developing metabarcoding assays for invasive insect pests. And this basically involves sequencing the entire trap sample at once and identifying all of the organisms in there. And I'm just going to show the same figure that Mark showed before. But basically the process is you get your trap samples, you take them back to the lab, you do a bulk DNA extraction on the whole sample. In our case, we're using a non-destructive method, so we can actually revisit the specimens if we do detect something and confirm them using traditional methods. You then enrich for a taxonomically informative loci. In our case, we target cytochrome oxidase 1 and use PCR amplification. You then label each sample with your unique molecular identifiers, pull them all together and sequence them on Illumina sequencing platform. You can then use bioinformatic techniques to clean up the resulting reads and assign taxonomy to these sequences, thereby identifying all of the species in a mixed trap sample. And this kind of gives you data that looks like this. Now, this was some validation work I did at the start of my PhD. And basically down the left, you can see all the species that were detected in these samples. I should mention these are mock communities I made up, which I put in some of these exotic species. And then down the bottom, you can see the samples they were detected in, and you also get an estimate of the relative abundance of each species. Now, with this data, you can compare it to your priority pest list to detect things you're looking for, but you can also compare it to your baseline knowledge of endemic diversity to incidentally detect other taxa that might have been missed by previous targeted surveillance programs. So during my PhD, I developed a metabarcoding assay and then did some field validation to see if it could detect spotted-winged Drosophila in a background of Australian insects. I did some trapping in a cherry orchard and a stone fruit orchard, which is a secondary host. And I caught about 1,000 Drosophila in the cherries, about 3,500 in the stone fruit, plus some other bycatch, and then also some larvae from crushing and floating these fruit. Then these are all combined by a week of collection, host, and treatment, and sequenced on an Illumina NovaSeq. So first off, in terms of results, I used a couple of different attractants I was testing. The first one was apple cider vinegar, which is basically the standard cheap approach used for surveillance in North America. However, one thing I found with this is looking at the samples successfully sequenced column is that only 1 out of 8 samples produced a sequenceable amplicon. And then with the synthetic lure, only 6 out of 8 samples produced a sequenceable amplicon. And this is, I believe, related to the specimen sitting in a highly acidic solution for up to a fortnight. And also the apple cider vinegar in particular is quite a complex matrix containing a lot of polysaccharides and things that can inhibit PCR reactions. So this third treatment was something I had developed, and that was the same synthetic lure, but with a propylene glycol DNA preservative in there, and also a dichlorvos insecticide cube to make sure the insects die and fall into the propylene glycol. And with this treatment, all 5 out of 5 samples were successfully sequenced, and it was similar for the fruit crushing and flotation. Now, looking at the graphs on the right, this is the number of individuals trapped in each sample, number of Drosophila individuals. You can see in the cherry orchard, the synthetic lure trapped the most, while the apple cider vinegar and the one with propylene glycol were about equivalent. And in the stone fruit orchard, the one with the propylene glycol and the dichlorvos insecticide caught more insects. On the bottom, it's species richness. So this is kind of a proxy for the selectivity of the traps. The apple cider vinegar, the one sample that worked, had quite high species richness. So it wasn't very selective, while the other 2 trapping treatments were quite similar. Now the fruit floats, while they collected a lot of specimens, they were mostly of the same species. The next thing I wanted to look at is, can we actually detect spotted-winged Drosophila in these trapped samples? So these were actually spiked with single specimens of Drosophila suzuki and its close relatives, which are also exotic, Drosophila subpulchrella and Drosophila biamapes. Despite seeing some deviation between what we expected in relative abundances from specimens and what we observed from the sequencing reads, we were able to detect all the single individuals across all samples. However, we did have one false positive for spotted-winged Drosophila, but luckily, because we had we used a non-destructive method, we could revisit these traps and confirm that wasn't actually a real detection. That was some form of laboratory contamination throughout the process. And this is something you really need to watch out for when you're using highly sensitive, high throughput sequencing methods. So this is just a quick overview of how metabarcoding also gives you all this biodiversity data. I'm not going to go into this too deeply in the interest of time, but basically a whole range of different species from 10 different taxonomic orders were detected in these samples. And you can see their presence, absence, and relative abundance changes between the cherry orchard, which is the inner heat map, and the stone fruit orchard, which is the outer heat map, and then the 4 different treatments used. So this was all work that I'd done in my PhD. And then after I submitted, we got some further funding to roll this assay out in a pilot surveillance program around Melbourne. And this was conducted in collaboration with Agriculture Victoria's Biosecurity and Agriculture Services, who were doing surveillance anyway as part of the National Plant Health Surveillance Program for spotted-winged Drosophila. And so basically about 58 samples were taken from a range of mostly pick your own berry farms around Melbourne. These were high risk sites. And we used this time this commercial Sentry lure and a BioBest Rosso trap. And again, having that propylene glycol, which I found preserves the DNA effectively. So just an overview of these results. So here you have each column is a different trap. You've got the relative abundance of the different species on the y-axis and the sample on the x-axis. So basically, the samples were dominated by 10 native Drosophila species. What we saw was especially the Scaptodrosophila lativittata, which is the purple, and Drosophila simulans, which is this kind of green color. And what we saw was the cherries, the strawberries, raspberries, blueberries, and blackberries, all these soft fruits, which are primary hosts of spotted-winged Drosophila. Their Drosophila communities were quite similar, while the citrus orchard was kind of dominated with some different taxa. So this shows that Drosophila communities are affected by the host type you put the traps out in. So the other interesting thing is because we get this other biodiversity data, we compared this to records on ALA and the Australian Faunal Directory, and we identified 5 putative first records for Australia. And these were non-pests, but there just hadn't been a previous detection in Australia. And these were one aphid, which was this aphid Pemphigus populividae, and then these 4 Scaridae flies. And because we used a non-destructive method, we were able to revisit our original samples, and our taxonomists could go through and pull out these specimens and confirm their identities using morphological features and DNA barcoding. So just to summarize this, basically I found that metabarcoding can successfully detect low abundance spotted-winged Drosophila in unsorted bulk trap catches, and it enabled the rapid identification of more than 18,000 specimens without requiring that laborious taxonomic sorting process. We didn't detect any spotted-winged Drosophila or other exotic Drosophila around Melbourne, thankfully, but we did reveal 5 putative first records for Australia, which non-destructive extraction enabled confirmation. And that's really, this non-destructive component, it's really a critical part of this process to make sure we adhere to kind of these concerns around detecting biosecurity related pests. So with that, I'd just like to acknowledge my PhD supervisors, Paul Cunningham, Noel Kogan, and Mark Blackett, as well as the range of different helpers I had throughout my PhD and over the course of this project. And I'll take any questions at that time.
Thank you, Alex. Very interesting talk. We already received one question from Elena McClelland. Her question is, awesome talk, Alex. How long were the trapped samples in the propylene glycol for?
So yeah, good question, Alana. I basically, so the propylene glycol was in the bottom of the traps in the field. And then they were collected every 2 weeks. For the stuff I did my PhD, when I got them back to the lab, I immediately filtered them out. But I believe for the second batch of stuff for this pilot surveillance program, they were taken back to the lab and put in the fridge in the propylene glycol for, I think it was a few more weeks before we got around to the DNA extraction.
Cool, thank you.
So any other questions? The audience can use the chat or raise your hand. Yeah, I have a question, Alex. So very interesting for the metabarcoding. Can it quantitatively tell you how many Drosophila suzuki in your back samples? For example, you have 100 different fruit flies, and after your metabarcoding, you know there is Drosophila suzuki, but do you know how many in this bulk sampling?
So in its current kind of form, it gives you an estimate of relative abundance, but that's quite biased because you've got these issues with like PCR primers can preferentially amplify different taxa. Also, What we've found through some other research is that DNA extraction in particular can be much more effective on certain species than others. So you do get a biased picture of the relative abundance at the end. However, it's generally in this like biased in the same way between different samples. So if you want to do things such as like measure where if one species is increasing or not, it works for that. But it's hard to get it back to like an absolute count to say like, oh, there's 5 spotted wing Drosophila in there. Maybe in the future, but it's currently kind of an unsolved problem.
Thank you, Alex. Let's see the questions. So next question is from Sonu. Question is, I might have missed it. What sequencing technique did you use for metal barcoding?
So as part of this work, I did most of the validation using an Illumina MiSeq. And then I used a Illumina NovaSeq for some of it. And that was just because I had a lot of samples for my other PhD chapters and I combined them kind of all together to get a cost-effective run. But we've used both of those platforms for metabarcoding so far. I'd love to expand in the future to trying it out on like portable platforms like the Nanopore MinION.
Good. And the next question is from Charles Robin. His question is, I was wondering whether there is active trapping for Suzuki incursions, and is it enough?
Enough. That's a good question, Charlie. Yeah, I think there's growing interest in having this trapping program. We're actually this season we're collaborating through the National Plant Health Surveillance Program again, but we're getting traps from other jurisdictions around Australia. Actually, I think I might have. Yeah, there we go. I hit a slide at the end for the next step. So James Mayno's modeling identified these priority industries around. We've got some other jurisdictions on board now.